to my channel. Today we will learn a new chapter, that is a lesson number 13 of the class 11th biology, photosynthesis and higher plants. Okay, first of all we will understand what is a photosynthesis. Photosynthesis is a conversion of the sunlight, carbon dioxide, water to produce glucose. It is an anabolic process which includes combination of the small molecules to produce a big molecules. Okay, so this is the reaction. In the glucose, the carbon bonds are present, are formed. Actually, to form this bond, energy is required. Photosynthesis is an anaerogenic process. What is the meaning of anaerogenic process? Means it is an utilize energy to do the reactions. Okay, now it is an anaerobic process, anaerogenic process. It is a redox. It is in the photosynthesis, redox reactions take place, means oxidation and reductions both take place. Now it is a physiochemical process, which means physical agent, light is involved and the chemical reaction take place. Okay, now site of photosynthesis. The photosynthesis take place in the chloroplast. In the chloroplast, there it includes stroma, grana, thylakoids. Okay, so first of all, there are two phases of the photosynthesis, that is light phase and the dark phase. Okay, or the light reaction or the dark reactions that is take place in the photosynthesis. This is the light reactions and dark reactions which is take place in the photosynthesis. Okay, now that these light reactions take place in the membrane system. This light reaction take place in the membrane system. Membrane system includes this grana and the thylakoids. Okay, this grana and the thylakoids, and these are the membrane system, and the dark reactions take place in this stroma. Okay, now further, the membrane system includes the photosystem. It is a complex protein and the pigment molecules. We will see what is the photosystem. Okay, it has a two compound, that is a reaction center and light harvesting complex. Okay, reaction center and light harvesting complex. This is the light harvesting complex and this is the reaction center. Okay, now there are two types of reaction center involves. Okay, two types of reaction center involves. First of all, the reaction center consists of the one molecules of the chlorophyll A. Reaction center consists of the one molecules of the chlorophyll A. It is called a photocenter. Primary reactions are performed. Okay, primary reactions are performed. Pigments are the molecules that, what are the pigments? Okay, first of all, what are the pigments? Pigments are the molecules that absorbs the lights. Okay, chlorophyll A has a capacity. Chlorophyll A has the capacity to absorb the wavelength of the 700 nanometer or 680 nanometer. There are, first of all, I told that there are two types of photosystems. Okay, the photosystem is comprised of the pigment molecules. These are the pigment molecules, and this is the reaction center. Now the photosystem are differ by difference in their reaction center. So two types of reaction centers are present. One is of a 700 nanometer wavelength, and other is of a 680 nanometer of the wavelength, which absorbs this wavelength. Okay, so two types of photosystem are present, in which first has a 700 nanometer wavelength photocenter, another has a 680 wavelength nanometer 680 nanometer wavelength photocenter. Sorry, that is reaction center. Okay, so the membrane system further includes photosystem. Okay, now the photosystem, there are two types of photosystem are present. Now first of all, that we will see after. First of all, this photosystem is consists of the reaction center and the light harvesting complex. Okay, this is the reaction center and this is the light harvesting complex. So first of all, what is the reaction center? It consists of the one molecules of the chlorophyll A. It is a pigment. Okay, so it is also called the photocenter. Primary reactions are performed in the reaction center. The pigments are the molecules that absorbs lights. Okay, chlorophyll A has a capacity to absorb the wavelength of a 700 nanometer or 680 nanometer. On the basis of this difference, the photosystems are two types. The one consists of the reaction center of a 700 nanometer, and other photosystem consists of the 680 nanometers of the wavelength. Okay, now the first light harvesting complex. What is light harvesting complex? Okay, are called antenna. They are also called antenna. These are also called the antenna. Consist of the hundreds of the pigments other than chlorophyll A. Okay, the chlorophyll B carotenoids, proteins are also present by absorbing light. Okay, by absorbing light energy, by light energy, electrons of the molecules get excited. Electrons of the molecule get excited and move far away from it. Okay, but to become stable again comes to the ground state, which releases energy, which is forwarded ahead. Okay, so first of all, light are being absorbed by this light harvesting complex. Okay, by the light harvesting complex, light are being absorbed, and the electrons present in this light harvesting complex get excited. Okay, they get excited, but to become stable they come again to the ground state. And first of all they release their energy. Okay, first of all they absorb, electrons get excited, electrons again come to the ground state, electron energy is released, and this energy energy is released by the one molecule get forwarded to the another molecule. Then first of all same process happen and forwarded to another molecule, same forward process happened, and finally the energy is forwarded to this reaction center. Then the first of all electrons of this reaction center get excited and it is transformed, that is through the electron transport system. Now where the photosystem 1 and the photosystem 2 are present. Okay, this photosystem 1 is present in the, now first of all this is the grana present in the chloroplast are divided into non-appressed regions, which is whose boundaries are not first of all attached to the another grana, and this is the appressed region, that is the middle part. There is appressed regions whose boundaries are attached to the another grana. Okay, whose boundaries are attached to the another grana. This is the non-appressed region and appressed regions. Now the photosystem 1, that is a photosystem 1 take place in the non-appressed region and the thylakoids. Okay, what is thylakoids? I have shown you in the diagram. These are the grana. Okay, these are the granas and these are the thylakoids. Okay, so first of all photosystem 1 take place in this non-appressed region and the thylakoids. Okay, and the photosystem 2 take place in this appressed regions. Okay, now the some informations regarding the phases of the photosystem, that is a light phase and dark phase. So first of all, light phase has the first, it is a light dependent, and the dark phase is light independent. It is called the Hill's reactions. Okay, it is called the Blackman's reactions, which is sometimes it is important for the NEET as well. So getting more information is also useful, so take it this into kind of information. So it is also called the Hill's reactions, it is called the Blackman's reactions. Then the Granal reactions or the thylakoid reactions, it is also called the Granal reaction. Thylakoid, it is also called the stromal reactions. Okay, photochemical process, it is called the biosynthetic phase. Why this biosynthetic phase? Because glucose is synthesis in this, and here photochemical process take place, production of ATP and NADPH O2. What is the outcome of the light phase? That is the production of the ATP, NADPH and O2, and the outcome of the dark phase is the glucose R synthesis. Now this is the what is this? This is a photosystem, and how the first of all what process will happen, I will tell you once again. That's first of all lights are fall on these molecules. This is a pigment. Okay, not chlorophyll A but other. Chlorophyll A is present in this reaction center. This is called the reaction center. This is a light harvesting complex. So when the light falls on this, electrons get excited and first of all to become stable again come to the ground state, and first to the energy which is absorbed by this molecule is released, and this released energy is absorbed by the another pigment. Then as such process take place, and finally the energies are transformed to this reaction center, and the electrons of this reaction center also get excited and again comes to the ground state, and the electrons, the energies are released in the first of all, but not, this is not. Okay, students make a mistake that the first of all they carry on this process, but this is not. That the light falls on these molecules, energy are transformed, and finally the electrons excited does not come to the come again to the ground state. They get excited and the electrons are transferred through the electron transport system. So this will see in the further. Okay, now this is one chart for that process I have told you. So it is useful for you to remember, that is events during the light phase, absorptions of the light by the pigment molecules, electrons of the pigments molecule get excited, electrons come back to the ground state by releasing the energy. Released energy is absorbed by the neighboring molecules, which in turn get excited and ultimately transform energy to PC, that is photo center. Okay, absorptions of the light by the photosensor and release the electrons and accepted by the electron acceptor. These electrons, I have told it is transferred to the electron transport system. First of all, it is accepted by this, that is electron acceptor, which is electron is released by this reaction center. It is accepted by the primary acceptor. This chlorophyll is also called the, first of all, primary pigment, because the primary reactions take place here and the first electrons are transferred to the primary acceptor. Okay, so remember it that the chlorophyll is also called primary pigment. Okay, so these are the events. Now the first of all, further this through the electrons are transferred, what happens what is the outcome of the transportation of the electrons? It takes place the first one, cyclic and non-cyclic photophosphorylations. What is photophosphorylations? We will talk further. Before talking about photophosphorylations, we will talk the first will take one information, that is redox potentials and the free energy. What is the redox potential? Redox potential is an ability to gain electrons. Okay, so from electron is transported from high redox potentials to low redox potentials, that is called uphill flow, and from low, that is redox potentials, to high redox potential, it is called a downhill flow. Similarly, the free energy which is present in this, so similarly the first of all, from one that is have the high free energy to the low free energy, if the electrons move from high free energy to low free energy, it is called uphill and downhill. Okay, but no, here it is different. Okay, sorry, here it is different. Video na pacho split kar lage. Okay, for this, okay, here if the energy is more, if the electron is transformed from the more energy to less energy molecules, then it is called a downhill process, and reverse, it is uphill process. It is reverse of the redox potential. Okay, now it is phosphorylations. What is photophosphorylations? Photophosphorylations in which addition of the phosphorus, it is called the phosphorylations. Okay, now we will what is cyclic? This is a cyclic photophosphorylations in which the energy is absorbed by this reaction center, electron is loosed. It has a first high redox potentials, then it has a low redox potential, it is primary electron acceptor, so it is called uphill process. Now the electrons which is accepted by the electron primary electron acceptor is transformed to the, first of all, this A1, okay, A dash, then the first of all FES, that is N sulfur protein, then it is FD, that is ferrodoxin, it is cytochrome B6F, then it is plastoquinone, and then the first one again it comes to, this is called a cyclic process. Okay, again it is coming to the photosystem 1. The cyclic process involves a photosystem 1. Remember that the cyclic process involves photosystem 1 having the wavelength of the 700 nanometer. Okay, now here when the first of all it reached to those cytochrome B6F, a proton motive force developed, and due to that force ATP, okay, ATP is formed. How the ATP is formed? An ADP, that is adenosine diphosphate. One ATP is added to it, it becomes triphosphate. Okay, proton motive force is developed here, the cytochrome B6F. Okay, now the first of all these terms which are written here, the first all have a first all look on this to remember. Right, this is first of all. So first of all, to get this kind of notes, PDF of notes, please join my Telegram channel, that is Patel Educations. I will provide you a link in my descriptions. Okay, the first of this is A0 is the special chlorophyll, that is a electron primary electron acceptor, that is a phytoquinone, then the FES is a electron sulfur protein, iron sulfur protein, that is cytochrome B6F, and this is ferrodoxin, and this is a plastoquinone. Okay, now this is non-cyclic photophosphorylation. What happens in the non-cyclic photophosphorylations? Okay, so first of all, energy is observed in the forms of the quantums. Okay, the non-quantums, that is photons. Okay, you have learned in your chemistry lecture, that is chemistry chapter 2, I think so. Yes, chapter 2. That is energy is absorbed in the form of the photons. Now one photons gives the transforms excites one electron. So the 4 photons are being first of all in the incident on the photo reaction center. So the 4 electrons are being loosed by the this photosystem too. Okay, first of all, remember this chart. This chart is very important for the NEET as well as schools exams also. Okay, many times the schools ask the first of all this non-cyclic photophosphorylations along with their chart. So please pay attention. So the first of all, 4 photons are incident on the reaction center, and that's why the 4 electrons are being moved to the primary electron acceptor in the photosystem to the first of primary electron acceptor. It is a pheophytin. Remember, pheophytin. Now these 4 electrons are passed to the plastoquinone and then to the cytochrome B6F. Now we learned the firstly in the cyclic process that at the for cytochrome B6F for proton body force is developed and the ATP is formed. Now further these electrons pass to the plastoquinone and then to the photosystem 1. Okay, now through the photosystem at the photosystem 1 also it requires energy to lose electrons. So 4 photons are incident on this reaction center, and again the electrons are moved. Now first of all it moves uphill process. Redox potential we have done, that is it is a uphill process, then it is accepted the primary electron acceptor, and then to the plastoquinone, that is plastoquinone, then it is iron sulfur protein, then to the ferrodoxin, and it is FNR. Okay, this FNR had 2 NADP plus plus 4 H plus 4 hydrogen. Okay, actually these 4 hydrogen pen is not working. So the first one 2 NADP plus 4 hydrogen. The first one 1 hydrogen has a 1 electron. Okay, so here it is 4 hydrogen. Okay, now 1 hydrogen has a 1 electron and 1 proton. So first of all 1 electron is used to remove this charge, it becomes NADP. Then first of all, then again 1 electron is 1 electron and proton is used to form H. Okay, so the first of all there are 3 plus 3 6 are used. First of all, what is happening, so understand it carefully. So 1 electron is used to remove this charge, so 1 electron is used. Then further 1 electron is again used to give the H. Now first of all here, so 1 electron and 1 proton is used, so 2 electron and 1 proton is used, 2 electron and 1 proton is used. So there are 2 NADP plus 4, so 4 electrons and 2 protons are being used, so 2 protons left here, so this is the hydrogen, so it is 4 used. Now how first of all it is necessary to understand that how from where these electrons are coming, which is excited by this. Okay, that is reaction center. So actually once it has some electrons it will lose, but there is will be electron deficiency here, and so which is first of all examined by the electron, that is oxygen evolving complex, which first of all which provide the hydrolysis of the water, not a hydrolysis but photolysis of the water. Sorry, it is a photolysis. First of all it carry the process photolysis of the water. It is called oxygen evolving complex. So the oxygen evolving complex photolysis of the water, at least what happens, 2 molecules of the H2O get broke down into the 4 electrons plus 4 protons, that is H plus. The electrons and protons are transferred in the form is represented in the forms of hydrogen and hydrogen. You remembered in the first of all your chemistry and the first 2 molecules. So overall the outcome of this non-cyclic photophosphorylation is that first of all it gives at ATP. Okay, it gives ATP, then the 2 NADPH, ATP to NADPH as well as the 1 molecules of the O2, ATP to NADPH 1 molecules of the O2, one non-cyclic photophosphorylations. Okay, now further if the first 4 photons are not provided here, electrons will excited, and if the electron will not excited, first of all these reactions stop. So so first of all So to produce one oxygen molecule, the first of 4 plus 4 8 photons are required. Okay, if 4 photons are not provided here, the process will not proceed forward, and the process stop here only. And the first of all here there will be no electron deficiency. So the first of all both these photons are necessary. So to produce one oxygen molecule, 8 photons are required. Now this is quantum yield, which I have discussed in the previously. So this is quantum number of the oxygen molecules released due to the one photon, that is the first one 8 photons are required to produce one oxygen molecule. So the 1 photons will produce 1 by 8 molecules of the oxygen. So the value is 1 by 8. Now the quantum requirement means the first one number of the photons required to release one oxygen molecule, that is value is 8. Now this is diagram of the first chloroplast and the first grana, and its lumen, and how the first of all electron transport systems are present in the first of all membranes and all. So first of all photosystem 2 is present in the first of all the membrane of the grana. Then the primary electron acceptor phosphorylating is present in the outer side of the first of grana, that is in the stroma it is present. This is the stroma, this is outer stroma, and this is the one grana. So it is present in the stroma. Then the first of the 4 electrons are transferred to here, and these 4 electrons are transferred to the plastoquinone, and now the 4 protons are added to it. So the 4 proton plus 4 electrons produce one, that is 2 hydrogen it will provide. So 2 hydrogen it will provide, so therefore it is PQH2, it will form PQH2. Now further passing through to the cytochrome P6F, it released that 4 the protons. Okay, now the concentration of the H plus the concentration increase here, which will feel further we will discuss after. Now the to the cytochrome P6F, it passed to the plastocyanon that is present inside of the grana, that is in the lumen it is present. Then to the photosystem 1, and then to the ferredoxin, and then it FNR, and that first of all the reactions we have discussed for earlier process. Okay, now the first of all due to the first of all addition of the H plus and the photolysis of the first of all water also take place here only through the electron oxygen evolving complex that we have discussed before. So the first of all here also 4 electrons are being used, but 4 first of all protons are present in this only. So the concentration of H plus increase. So the first of all the H plus wants to move out of this grana, and out of when they move, the first of all they release energy, and that energy is called a proton. Actually the energy is force. Okay, so that is the proton motive force. Okay, that is the proton motive force, and due to that force the ADP the ATP phosphorus is added to the ADP and forms ATP. Okay, here the concentration of the H plus is get low. That's why the first voltage difference in their voltage, and this such due to which the proton motive force is developed and all. Okay. Now the orientation of the chloroplast, when the high intensity of light is fall on the first of plants, they arrange in the perpendicular to the that is surface. It is a perpendicular arrangement. When the low intensity of the light is incident on the plant, then they arrange themselves in the first of all parallel form, and the first of all normal they roam in the first of all these types of take place. Okay, so this is the reaction of photosynthesis. Now the first of the dark reaction. So what is dark reaction is called a misnomer. What is a misnomer? That is means understand grammar, that is to say something else or to perform is function is something else. Means the dark reaction is only to say, it is also performs in the light as well. Okay, it is called a biosynthetic phase, the formation of because of the formation of the glucose. Now this is the Calvin cycle or the C3 cycles. It take place in the first of C3, C4 AM plants. Then the first it take place in the cells, that is mesophyll cells or the bundle seed cells. Okay. Now in the organelles, that is a chloroplast and stroma. And the first of Rubisco. What is Rubisco? Rubisco is the enzyme, Calvin cycle enzymes. It regulates the speed of the C3 cycle. Then the first of phase, it is a pacemaker enzymes of the C3 cycle. Most abundant proteins in the air now found in the stomach. Now the first of it is bifunctional. Then the first of all its cofactor is the first one magnesium 2 plus. Its cofactor is magnesium 2 plus. What is the cofactor? Okay, the first one what is enzymes? Okay, enzymes are mainly made up of a protein. Now the enzymes are the enzymes which are not made of a protein are called a cofactor. Okay, understand the coenzymes here only. So the first of these are also the enzymes which are not made up of a protein, that is called a cofactor. If these cofactors are the organic made up of organic compounds, then it is called a coenzymes. Okay, now the first of all activities of the Rubisco. Activities of the Rubisco, it first of all first of all carboxylase and the oxygenase process. It first of all RUBP, that is ribulose 1,3 bis phosphate. The first of all it carboxylize the RUBP by addition of the carbon dioxide. This process is called C3 cycle, and the first of all addition of the RUBP plus oxygenase, that is called C2 cycles. Okay, it is a waste process because no ATP is formed. Actually, the ATP is used in the C2 cycles. Now we will further discuss the C3 cycle. Take a gist of the first one C3 cycle. It is Melvin Kelvin. Okay, studied algal photosynthesis. Okay, algal, it was studied first of all for the algal photosynthesis, cholera. There is another is then the first one is experiment. A specimen used towards cholera, then the radio isotopes. That is then the first of all technique used in this experiment was the first of all autoradiography. Okay, autoradiography, that is a technique used in this. Now the first of all found for the first stable product in the first of all C3 cycle is a phosphoglyceric acid, that is which consists of 3 carbon. That's why it is called a C3 cycle. Okay, also help in the finding the remaining reactions of the Kelvin cycle. So now the Rubisco. Okay, the first of all Rubisco is called the hero of the Kelvin cycle, occurs in the stroma of the chloroplast. And the Rubisco actually has the binding affinity for the carbon dioxide or the oxygens, but in the condition of the carbon dioxide and the carbon dioxide concentration and oxygen concentration same, firstly it has a more affinity for the carbon dioxide, so the C3 cycle mainly occurs. Okay.